Hello and welcome to this video on why your blood does not turn into an alkali or acidic solution every time you breathe. This is all down to a principle in chemistry known as a buffer. Buffers act as an acid when in the presence of an alkali and act as an alkali when in the presence of an acid. How these chemicals work is something of a quirk of the table of elements and just what makes an acid and a base. This also leads to it being an incredibly powerful phenomena in the right circumstances. Before explaining what a buffer is, we need to touch on the periodic table so you can have a little understanding of what makes an acid and a base. Each column of the periodic table on the sides, or non-metallic elements, corresponds to a free electron position. This means everything under lithium on the far left has one free electron space. Everything under beryllium has 2, boron has 3, carbon 4, and so on going to the right. The metallic elements are an exception to this pattern. When these are combined with hydrogen, we start to get an acid or a base. If the compound, as per the simple definition, donates the hydrogen that has filled this empty space, it is an acid. When it receives the hydrogen, it is an alkali or base. A buffer will change between these two states, as it is either a weak acid or weak alkali, depending on the circumstances. The more strict definition is that a buffer is a solution containing either a weak acid and its salt, or weak base and its salt, which is resistant to changes in pH. With this technical knowledge, we can see how a buffer works now. Buffers keep a very stable pH in a given solution. They effectively counter the change from an acid or a base, neutralizing it. For any buffer, there is a particular range in the pH spectrum in which they will work. If you get out of this range, the buffer is overcome. If you are within that range, it will neutralize it and the pH will not change. You can begin to understand just what relationship your weak base and weak acid have via something called the Henderson-Hasselbalch equation. This uses a series of given symbols, which can be translated into specific values. Your ApH, or pH at which your buffer is working, your pK, which is a constant value for any given particular thing you're looking at. Generally, it is the acid. Log is a function, and it applies to the molar concentration of your conjugate base, which is your A-, which is put over your molar concentration of your undisassociated weak acid, or HA. Together, this should give you some idea of the nature of how effective your buffer is going to work in a given situation. At half the value given from this equation, your pH should equal your pKa. Now that we've had a look at the theory behind this, let's look at it in practice, specifically how the buffer works in a solution. If you were to make a buffer solution from sodium acetate or sodium bicarbonate if you can't get your hands on sodium acetate, and acetic acid being effectively white vinegar, you can begin to see what's happening. When you combine these two, you get a slight reaction in the case of bicarbonate and vinegar. This is the two neutralizing each other. That's what that reaction is. If you were to use the sodium acetate, the acetate would cause a slight breakdown. The sodium acetate breaks down in such a way that it can neutralize. That is, the acetate part will separate from the sodium part, and the acetate will bind to your acetic acid, leaving only sodium, which is a salt, behind. Because the acidic effect of the acetic acid is countered, the pH does not shift. If you were instead to combine the acetic acid with a strong base, you would get more of a reaction. That's roughly what you're seeing if you combine it with sodium bicarbonate. The reason you see the pronounced reaction between the bicarbonate soda and the acetic acid, as opposed to the acetic acid and sodium acetate, is that the bicarbonate soda is a much more alkali solution. If you were to have a more closely matched pH range, this is where a buffer would come into play. Buffers generally only work over a relatively narrow pH range. There are some that are much broader, citric acid being an example of this. The reason citric acid works at a much broader range is that it has 3 pK values. This means that there are 3 points at which the pH can be modified, and it functions as a buffer. If you were looking to achieve a wider coverage and you weren't able to find a single buffer that had a wide enough range, you can in fact combine multiple. If you have some overlap between different buffers, you can have them working in conjunction to try and keep the pH steady. By having them overlap, even when one's not working as effectively or has diminishing function, the other should begin picking up. This means you can have a buffer that runs from the very low pH measurements through to extremely high pH measurements. That's everything from your extremely strong acids all the way up to your extremely alkali solutions. This is extremely useful in research. Biologically, not so much. Despite that, buffers do find a role to play in most biological research, in one way or another. 2 examples are PBS and TAE. PBS is phosphate buffered saline. This is a supplement for water that has the right salts included, but it also has that buffer in it. The buffer allows it to work between 7.3 and 7.5 on the pH scale, exactly the right range for biological testing. It's often used for cell culture, immunoassays, and protein purification for this exact reason. TAE is a little bit different. It stands for tris acetate EDTA buffer. It works between 8 and 8.3, a much higher measure on the pH scale, and far more basic. That's because it's not trying to keep biological functions going. Rather, it's often used for analytical research. Big functions are for agarose gel electrophoresis. What you're doing here is you're trying to separate out the various parts of the DNA and RNA that you're sampling to get them to break into bands based on their size and weight. Generally speaking, it's primarily used for nucleic acids. There are other buffers used for protein that does other kinds of blotting. If you didn't have the right pH for this to happen, you'd find that the DNA or RNA began to unravel or unfold in the wrong ways, and would get stuck in the agarose gel. As a result, this would lead to it not moving in the right way, and you would not get nice, neat, clean bands. Let's now apply what we know of buffers to the blood-based carbonic acid bicarbonate buffer, the very thing we introduced this video with. Human blood contains a buffer of carbonic acid and bicarbonate anions. These keep your blood between 7.35 and 7.45. If your blood goes below 7.35, this is called acidosis. Diabetics are prone to it. It can cause significant changes to the body and lead to death relatively quickly. If it goes above 7.45, this is called alkalosis. If you've ever had an attack of hyperventilation or similar, this is what you're experiencing after a while, alkalosis. In order for the human body to function properly, it has to keep blood in the right range. One of the reasons this happens is that as you breathe in oxygen, you exhale carbon dioxide. The carbon dioxide can bind to the red blood cells in lieu of oxygen, but this is often not the case due to different concentrations of each particular element in your blood. Instead, the carbon part of this is dissolved in your blood as carbonic acid. When it gets to the lungs, it turns back into a gas and you exhale it. If you aren't exhaling enough, you would get a buildup of carbonic acid in your blood, and acid in your blood would be a bad idea. As a result, the bicarbonate is there in order to counter its effect and bring your blood back down to that narrow pH range. That's a 0.01 pH range in which your blood can function the way it's meant to, and that keeps your body going. The benefit of a buffer is that it keeps the pH of any given solution relatively constant, not just biological organisms, cells, research samples, or anything, but in chemistry overall. In very simple terms, it shifts the reaction in the opposite direction of the pH added, and this keeps it reasonably stable, again within a narrow range depending on what sort of buffer you're using. For the most part, it's not discriminatory in how it functions. For example, if you were to drink a bottle of something acidic, assuming it could get to your stomach and begin being absorbed into your bloodstream, this buffer would stop it from shifting the pH too far. Conversely, an alkali solution has some of the same effect if it can get absorbed. In many cases, your body would reject it and you would have some sort of way of disposing of it, whether that is vomiting or something else. This is also why, as we've mentioned in other videos, an alkali or acidic diet will do nothing for you. Certain lifestyle changes or detox diets have no benefit, and more, simply because the human body, like most organisms, is complex enough that it can counteract many of these changes that you try and bring on. In fact, most of the time, the only reason the body's pH changes drastically away from that 7.45 or 7.35 is that there's some sort of larger scale pathology going on, something that's causing drastic change in your body's ability to function. This could be kidney damage, lung damage, issues with the retention or release of certain chemicals within the body, such as calcium. If your body begins dumping these materials out of your bones, your kidneys are behaving incorrectly, or your lungs aren't working, this is generally not down to diet. Rather, it's a disease of some sort. If you'd like more information on how any of these things are either working or not working, as claimed in much of the pseudoscience and woo that is out there, we have some videos linked in the top right hand corner and in the description below. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.